Let me introduce our first speaker, Sean Ekins. Sean is CEO and founder of Collaborations Pharmaceuticals, that is focused on rare and neglected disease drug discovery using machine learning and AI approaches. After his postdoc at Eli Lilly, Sean started his industry scientific career with Pfizer, then later with startup pharma and software companies such as Concurrent, GeneGo, CDD and Phoenix Nest. As he has also served as scientific advisor for rare disease foundations. He has authored or co-authored more than 300 plus peer-reviewed papers and book chapters. Sean's topic of presentation is applying AI in a small drug discovery. Sean, the floor is all yours. You have 25 minutes for the talk, Sean, and at 22 minutes, I will inform you to wrap up, if that's okay. Back to you, Sean.
Thanks very much, Shyam. It's great to be here. Great honor to be invited, and just like to thank the organizers for putting this together. It's been absolutely stimulating watching and listening to the talks today. So I'm going to be giving obviously an overview of what we've been able to do since founding this small company. So it's going to be quite different to most of the talks today, which have been predominantly academic, with obviously the exception of the keynote coming from Amgen. So we're a pretty small company, and the folks on this slide are essentially responsible for most of the work that I'm going to present. So as you can see, there's about a dozen or so of us currently, and we develop various AI, machine learning technologies and software, which we apply for various drug discovery and consumer product applications. And probably what's unique about us, I would say, is that we haven't been funded by venture capital or angel funding. And we've initially relied heavily on funding from the NIH and Department of Defense. So lots of grant writing, and that's enabled us to bring in about $23.5 million so far, with obviously collaborators on various projects since 2016. Pretty unique way to fund a small company. We have labs. We're actually a real company. So we have 3 labs and an office on the campus here at NC State University. Now, we're not actually affiliated with the university, but we definitely take advantage of great collaborations here and access to amazing students and faculty. And over the years, we've been able to produce a number of patents and obviously publish quite widely, as you're going to see in a few moments. So my focus is today, obviously I can't talk about every single thing that we've done since founding the company, but I want to try to give you some ideas of what's possible using these technologies, and also maybe try to inspire other people to think about starting their own company that's driven by AI approaches. So at the heart of what we do as a company, we're essentially a 3-in-1 organization. We build our own software and license that to companies, both for predicting properties and obviously toxicity is a big one, which I'm going to touch on a little bit later. We also use these technologies to help drive our pipeline, so find molecules and optimize molecules. And then thirdly, we're a contract services organization. So pharmaceutical companies and consumer product companies come to us with their challenging problems, and we work on solving them using our technologies. And that's so far enabled us to bootstrap the company. And we've been able to use and start off initially with obviously traditional AI approaches. And these are obviously tools that you've seen quite a bit of today already. Tools that will literally take a data set, whether that's a bioactivity or a physicochemical property, for example, and then allow you to build a predictive model and then obviously generate predictions for new molecules, whether those are from a virtual library or an available library of molecules, for example. They interestingly don't necessarily allow you to create anything new, but they allow you to mine the data and find potentially new molecules that have new uses. And I'm going to try to show some of those examples after this slide. And then the second approach, which you've seen a little bit more of today in the more recent presentations, is more of the generative AI, where you can have these technologies that will learn from the data sets, whether these are large language models or other types of graph-based approaches, for example, and then they'll enable making decisions from that data and really going beyond the data to propose new novel molecules in this case. And what's really interesting, I think, as a small company applying these types of technologies is that we're literally at the cutting edge of using these tools. And when we see what the big pharma are doing with obviously much more investment, way more investment than the 23.5 million or so that we brought in from NIH and DoD, it makes you realize that big pharma has massively underinvested in AI to date. And so to some people that may seem controversial because they're throwing money at it hand over fist. But actually, we've had several companies come to us and actually tell us that they're a decade plus behind. So I think there's still a lot of opportunity out there for these types of companies to invest in this space. So the types of tools that we initially used, started off with algorithms like Bayesian algorithms, have been around for a long time, random forest, etc. And over the years, we expanded to look at many different algorithms in parallel, both for classification and regression, and then obviously in more recent years, more sophisticated tools like convolutional approaches and LSTMs and transformers. So a whole mix of different algorithms. And as I'm going to touch on a little bit later, picking which algorithm to use for a particular data set is in itself a challenge. So initially, we were really focused on trying to repurpose molecules. As a tiny company, really what we were doing was design, buy, test, analyze. We weren't even thinking about making molecules because it was just way beyond what we were able to do. Initially, as a small company, we didn't even have a lab. So we were using technologies and building technologies so we could screen through libraries of FDA-approved drugs, etc., pick out molecules, and then send them off to collaborators and perform the testing. Pretty simple, basic type of approach, one target at a time or one phenotypic assay at a time. And this ultimately led us to put together enough test cases that we could say, okay, let's demonstrate this. Let's produce some software around these types of algorithms. Let's bundle not just one algorithm, but currently 15 or so different machine learning algorithms. So we've now created a software called Assay Central, which we license to companies so that they can bring in their own data set, curate it, clean it up, and then build lots of models in parallel and use them for predictions. So what you're going to see over the next few slides are really things that we've done pre-developing these technologies. And they obviously led us to develop the tools that we have now been able to license to companies. So one of the really early examples that we published back in about 2015 was around the Ebola outbreak of 2014. Obviously, nowadays we think of major virus outbreaks like SARS-CoV, more on that later. But back in 2014, there was quite a big Ebola outbreak. And at that time, I was interested in trying to access data sets to build a machine learning model that we could then use to try to identify new antivirals. And one of our collaborators, Peter Madrid, at the time at SRI, he had published in 2013 a data set that we're able to get a hands-on and build a Bayesian machine learning model that we've then able to use to virtually screen a library of about 2,500 molecules in the microsource library. And that then enabled us to rank the molecules, and we selected 3 of them, quinacrine, tellurone, and pyrinaridine, for testing with a collaborator in vitro. And we're able to find that all 3 of those molecules that hadn't been tested previously were actually nanomolar actives in this in vitro assay. And that was surprising for a number of reasons. Obviously, I don't have a lot of time to go into this today, but one of the molecules that came out of it was an antimalarial that's used as a combination with another drug called artesunate, and that was approved by the EMA several years after this publication. And we were able to use this preliminary data, literally this one graph, to get an R21 from NCAT. So we're very grateful to them. And that really kicked off work within our company in terms of evaluating this molecule, not only for Ebola, but also against other viruses. So over subsequent years, we've been able to use this molecule that was identified through machine learning and characterize it to try to understand the mechanism. So we showed that it's lysosomotropic, it accumulates in the lysosomes, changes the pH. It's a potent inhibitor of lysotracker red, submicromolar. And subsequently, we also performed some docking to evaluate a number of targets, one of which was the Ebola virus glycoprotein. We followed this up with doing microscale thermophoresis to look at binding and showed that it's a pretty potent low micromolar binder to this target. And then we also looked at how the molecule may interfere with access of the virus into cells. So we showed with the pseudovirus that this molecule pyrineridine tetraphosphate was able to block uptake. So really interesting, over the years, we've been able to really fill out what's understood about this molecule that, as I said, is traditionally thought of as an antimalarial. Obviously, our goal with the NCATS funding was to try to move the molecule along as far as we could. And so we were focused on trying to generate PK data and ADME data. And we were able to show pretty quickly that it had a very long half-life, about a week. And we could dose this IP and have pretty good PK information. So this then enabled us to do a single-dose IP study in mice and showed that we had 100% survival. And we could do a time assay as well, so providing the drug at windows after dosing with the actual Ebola virus, and showing that we could have protection up to 24 hours. So we were able to look at plaque assay as well as PCR to assess the viral levels. And this work was published by Tom Lane in our lab, and he's actually been responsible for most of the work on pyrineridine during this period and since. And ultimately, this enabled us to obtain an orphan drug designation from the FDA as well in 2019. Following this, we were able to characterize the molecule in guinea pig, show that we could dose the molecule a single dose, 300 mg per kg, and we had some protection against the Ebola virus. It wasn't as convincing as in the mouse, and we think this is primarily due to the increased metabolism in the guinea pig. And additional studies with collaborators at NIH also identified that pyrineridine was active not just against a single strain of the virus, but against 3 strains, Kikwit, Makona, and Mayinga. And in these assays, we're able to show an IC50 of around a single micromolar. The next stage for this project is obviously to get it into non-human primates. And for that, we have to obtain several $1 million of funding. And we're currently putting grants in to try to get that work done. What was interesting for us around 2020, we could not have predicted that there would be a major outbreak of a new virus that ultimately became SARS-CoV-2. And we were quite excited because remdesivir, another Ebola drug, had been shown to be efficacious against the virus. And so we wanted to try to see as quickly as possible if pyrineridine could have activity. And so we collaborated with a number of groups, both in the US and outside of the US, to try to test the molecule. Initially, we were able to show activity in cell lines. Interestingly, pyrineridine is not active in vero cells. And so we found activity in A549 cells. And then ultimately, we're able to test this molecule in a mouse model with a group in Brazil and show that it was efficacious. But kind of exciting for us was it had an impact on inflammatory cytokines. And so it was basically an anti-inflammatory molecule and decreased cell infiltration. And after the fact, we also showed that it was a inhibitor of PL-PRO in vitro. So this single molecule initially identified for Ebola appears to target totally different targets in different viruses based on the data we have. And a pharmaceutical company, Xingpeng Pharmaceuticals, actually took this combination, pyrineridine and alloxunate, which they make for malaria. They took it through a phase 3 clinical trial as well in humans. At about the same time as we were working on pyrineridine for Ebola, we had done a virtual screen again with a data set, this time for a parasite called T. cruzi, or ultimately resulting in Chagas disease. And this was with collaborators at UCSD. Here, there's a photo of Jair and Diane at UCSD that did a lot of the work, along with Laura Isabel McCall, who was a student at the time. And in this study, we looked at building a model with about 4,000 molecules from the literature, and then we screened a library of about 7,500 molecules and picked roughly about 100 of them for testing, of which roughly 17 or so had good activity and 5 were in the nanomolar range. And then we're able to test the molecules in vivo in animals in the acute model, and we showed that pyranardine had really good efficacy. And again, we used this in vivo data to obtain the orphan drug designation for this particular use. And that also led us to obtaining another grant from NCATS, which was a UG2, UG3 grant, which funded Jair and his group to do more in vivo efficacy work and also PK. And in this case, we were trying to look at different strains and ultimately try to show whether there was any efficacy in the chronic model of Chagas disease. So what we ultimately found using now we've progressed from IP dosing the drug to dosing the drug orally. We're able to show efficacy still in acute models with a different strain of the parasite, but unfortunately we weren't able to show efficacy in the much longer chronic model that basically goes on for about 6 months or so. And so we think that there's still some way to go in terms of optimizing the models here in terms of the strains. We might have a strain-dependent effect of the molecule. So interestingly, sort of a high-level overview of how we went from machine learning to identify molecules all the way through in vivo. We've also done similar types of virtual screens for other targets, including GPCRs. We've done some work that's highlighted briefly on this slide with a couple of recent publications. Again, this work was led by Anna Poole, who was in our group at the time, and working with Ken Jacobson at the NIH. And this work basically demonstrates that we're able to find new uses for FDA-approved drugs, such as febuxostat and paroxetine. They seem to have activity in radioligand binding assays for a number of adenosine receptors. And then a number of clinical stage molecules we've identified having activity in some purinergic targets as well. So definitely we've tried these technologies in terms of phenotypic screens as well as more target-based screens. And then a more recent example was at the request of an academic, Keith Pennypacker, University of Kentucky, who had identified a couple of chemokine receptors, CCR3 and 4, as being involved in stroke. And he wondered if we could find an FDA-approved drug that might be an antagonist. And so we went through the literature trying to find molecules, curated a couple of data sets, built machine learning models, and then ultimately used those to screen a library of FDA-approved drugs. We're able to find a number of kinase inhibitors that also have activity against CCR3 and 4. And then more recently, we've shown that this one particular molecule that we focused in on had activity against CCR5, which is also a target for HIV. So we've done pseudovirus work to show that the molecule actually blocks entry as well. So really interesting how we've been able to take this basic machine learning approach and really go after different types of targets. And obviously, we've also looked at enzymes, an example being butyrylcholinesterase, an important enzyme for a number of CNS diseases, including Alzheimer's disease, as well as potential toxicity target through molecules in the environment, pesticides, etc. And there isn't currently a selective butyrylcholinesterase inhibitor that's FDA approved. So we looked at different machine learning approaches to try to come up with a selective molecule that wouldn't hit acetylcholinesterase. And so we built models with a couple of thousand molecules that were curated from the literature, both for butyrylcholinesterase as well as acetylcholinesterase. And we came up with a number of machine learning approaches like random forest, deep learning, and an approach called contrastive learning that would hopefully enable us to try to produce selective molecules. Once we validated these models, This was work with a postdoc in our group, Kamal Ozalt, and we did a virtual screen of about 5 million commercial molecules, selected molecules for testing in vitro, and then we purchased about 20 molecules for testing, and then followed up with dose response data. Ultimately, we're able to get about a 35% hit rate, and one molecule had an IC50 of 2 micromolar, selective against butyrylcholinesterase, and was comparable to a control molecule, rivastigmine, with an IC50 of about 0.5 micromolar. So fascinating approach, trying to find molecules that are selective. All of these are really going after trying to find bioactive molecules. These are great examples, but there's also a growing amount of data out there for trying to understand the properties of molecules, like ADME-tox properties, for example. And although these data sets are nowhere near the amount that a large pharma would have, they are enough information to build pretty convincing machine learning models that we can take advantage of. And I'm just going to show one example of a recent publication from last year. We were curating data sets for not only rats, but also mouse, Daphnia, and various fish species in order to build both classification and regression models to predict LD50 and LC50. So these models then we can combine and make available in a tool we've called Megatox. We've done a bit of work as well in terms of pulling together larger collections of models, and this has led us to look at different large language models. And as I mentioned earlier, one of the challenges is to try to come up with a way to hone in on which model to use with which particular data set. And so we've looked at this, and a recent publication goes into this called the Goldilocks. So I definitely recommend checking that out. And then the last example I'd just like to briefly provide is our work around using generative AI. So we've obviously built on some of the tools that are available. We focused in on using an LSTM type of model and plugged in the various models we built for both bioactivity and ADME-tox properties. And we use a couple of different ways of being able to generate molecules. We could think of this approach as being an inner loop and an outer loop, where, for example, if we're trying to get selectivity, we would have various thresholds for the targets and off-targets of interest, and obviously go through this loop selectively until we come up with molecules that pass our criteria. And one example I'm just going to show is currently a manuscript that's been submitted. So this is unpublished as of now, where we started using psychedelic molecule psilocin with models for various 5-HT receptors, trying to find molecules that were agonists and selective. And ultimately, the model that we generated came up with 10 molecules that we're able to synthesize, and we performed the in vitro testing. And ultimately, one of these turned out to be a 5-HT2C selective agonist with an EC50 of about 12 nanomolar. And for reference, a molecule like lorcaserin, which at one time was FDA approved, has an EC50 against this receptor at 9 nanomolar. We don't think this is a perfect molecule that we've come up with, but we have been able to dose it IP in mice and shown that we can achieve good brain levels. So a real-world example of us going beyond just trying to find molecules and repurpose them, but actually trying to design, make, and synthesize the molecules. Obviously, much more expensive and time-consuming. Our interest in this area definitely has kind of taken off in terms of trying to avoid molecules that have psychedelic effects. And this led us to going out into the literature, trying to find data sets, both in vitro and in vivo, that we could then model to predict whether a molecule is going to be psychedelic or not. And a recent publication in ACS Chemical Neuroscience highlights this work. A great example here of just going into the literature and trying to manually create data from books like Shulgin and Shulgin's books, for example, where he'd actually made molecules that were psychedelic or not, tested them on himself and friends, and then scored them. So we were able to take that information and build classification models that we were then able to validate with additional molecules that were not in his data set. And so these models, both in vitro and in vivo, we've used to predict other molecules, as well as molecules with mouse head twitch data. So a starting point really to try to come up with the next generation of models to predict psychedelic effect. Obviously, there is great opportunity with these kinds of algorithms that can design new molecules, but there's also a word of caution. And one example led to a publication a couple of years ago that received a little bit of visibility and ended up on Netflix. And that was where we were trying to generate molecules at the request of a conference to show the potential for misuse of AI. In this case, we were designing new VX molecules using a couple of models we had for acetylcholinesterase inhibition and rat LD50. Obviously, this is not ideal. We didn't make the molecules, but it just goes to show that these kinds of technologies can design plausible molecules, and the potential here for misuse is obviously great. So just to summarize, obviously, there's a large amount of information on bioactivity for various targets in the public domain. This is ripe for curation and modeling. Hopefully, what I've given today is really just a taste of what we've been able to do with very small amounts of funding in general terms. I think AI enables us to move definitely faster, definitely saves us a lot of money. We're doing this pretty cheaply, and obviously, we're able to reduce animal use. The challenges, obviously, are that we have to go much further. We have to take these hits, these molecules that are active in animal models, all the way to clinical trials, and that's still massively expensive. So designing is certainly cheap, finding molecules is cheap, testing them or synthesizing them is just a huge bottleneck. And the final challenge I think we face is obviously bigger companies still want clinical stage assets. So until they get to the point where they need earlier assets, we are still going to have this valley of death to sort of bridge. So I think obviously AI is another tool in our toolkit. And for us, it's been obviously impactful. For that, I'd like to acknowledge the NIH, who primarily funded all of the work that I've shown here. And obviously our collaborators on Ebola that are shown on this slide. Way too many collaborators, unfortunately, to acknowledge, but really grateful for their impact. And obviously our collaborators on COVID, which were global, shown on this slide. And finally, my team who've been involved. Most of the work that I've shown you has involved Tom Lane, Josh Harris, Patricia Vigneault, and to some extent also Thane Jones and Scott Snyder. If you'd like to learn more about generative drug discovery, we've put together a book that should be coming out later this year. I'd also like to thank our other collaborators that haven't been acknowledged. And if you have any AI-focused manuscript, please submit them to Gen Biotechnology, Drug Metabolism Disposition, and AI Life Sciences. Thank you.
Thank you, Sean, for an excellent talk. I'm trying to see if there are any questions. No questions so far. Maybe I can ask one question, Sean. The molecules that you showed, particularly for Ebola, the pyronaridine, I assume, is that manufactured at this point of time, or is it more still in the lab development stages?
Well, it's actually an approved drug in Europe. It's used in Africa. It's used as an antimalarial in combination with artesunate, but it's not used as an individual molecule. So what we were showing was the individual use of pyronaridine tetraphosphate is actually where we're seeing the antiviral activity. And prior to us showing this, no one had previously shown that there was an antiviral activity with this compound.
No, the reason to ask was also because, at BARDA, there is an interest in Ebola therapeutics, vaccines as well as Ebola therapeutics. And I thought the data that he showed on this particular molecule was interesting and would be of interest to the BARDA, CBRN group, basically. And I'm happy to touch base with you.
Yeah.
Later on this. I'll follow up with you on this.
Yeah, that would be great. Thank you. Yeah, we've definitely presented it to BARDA over the years. I think when there was the COVID outbreak, we presented this several times. And really, to be quite honest, we got no interest from any of the organizations we presented to, because I think they were looking for novel molecules and less on the repurposing side. So, I mean, there's definitely pros and cons of repurposing. And yeah, we were certainly really interested to see what initiatives were out there. But as a small company, some of the initiatives required us to put up funding, and we certainly didn't have the wherewithal to do that. And I think that was one of the challenges of some of the BARDA initiatives. was that there was a significant investment that would be required from our side. And that's just impossible for tiny companies. So it just was unrealistic. But yeah, certainly open to ideas. Thank you.
And based on what you just presented, all the other molecules also, especially in the GPCR modulators or beta-alkoholinesterase inhibition, is the focus to look at already established molecules and repurpose them, reposition them? It seems like your AI tools that you are using are predominantly directed towards that effort rather than developing new molecules. Is that a fair statement?
Well, what I was trying to do, and I probably wasn't very clear about it, which I'm happy to clarify. So that's a great question. Initially, we started off heavily on repurposing, heavily on the traditional AI, just trying to mine data sets. And then over time, we've sort of moved more into designing new molecules, so more de novo design. And I think that's where we are today. We're more on that de novo design side. Yeah, we do still do a lot of the sort of screening through data sets, trying to find hits. And that obviously was a great foundation for the company. But we had so much feedback from big pharmas just saying, I would say about 4 years ago, that they were only interested in novel molecules. They weren't interested in repurposing. And that was really the impetus, I think, that drove us to then go after doing more of the generative design. And then obviously we had to go off and try to get grants that would fund us to do the synthesis. So, we've been fortunate to get grants from NIGMS, NIEHS most recently, and before that NIDA, that have allowed us to do more of the generative design. So trying to come up with molecules for opioid use disorder, for example. And most recently, we've been able to leverage the butyrylcholinesterase and acetylcholinesterase inhibition work to try to design molecules as potential reactivators that could be used for pesticide and chemical weapon poisoning. So it's definitely really evolved, and there's been some serious pivots in the company in terms of, where we've taken the technologies. But yeah, happy to, follow up with anyone if they have questions, if you want to drop me emails or whatever.
I'm just quickly following up on some of the open questions that are there. I'll ask a couple of them, and maybe if you can stay online and respond to some of them later.
Yeah, thank you.
One of the questions was, instead of synthesizing compounds, do you have good luck purchasing existing compounds that have a decent structural similarity to your virtual hits?
Yeah, we've definitely have tried that as well. I mean, I think the most convincing, this is just a really philosophical question in terms of how do you really want to prospectively validate your models. I've always been interested in how to validate the models effectively that are convincing. And you can never really win over people unless you actually design novel molecules. Buying them is really a cheap way to do it, and I'm fine with that. But people are not going to be really truly convinced until you actually design truly novel molecules that are not available from anyone else, have never been published, have not been put into a patent. That's the only way we're going to convince people. And then you're just going to get into the minutiae about how similar the molecules are to things that exist. It's never ending. So you can't win, to be quite honest, because, someone's going to find a method, some Tanimoto similarity or whatever, that, it's similar to this existing molecule. It doesn't look anything like it. But so there is definitely it's cheaper to do that. But, the most convincing thing is really to make the molecules.
If you can stay online, Sean, and answer some of the rest of the questions.
Thank you.
That will be great.